This is the third lesson of Chapter 8, where we're going to continue looking at a more depth view of photosynthesis. And we left off with the light reactions, so now we're going to study the Calvin cycle. Recall that from the end of the light reactions lesson that ATP and NADPH were produced in the stroma of the chloroplast, and they're now available to fuel the Calvin cycle. In the Calvin cycle, we see the influx of CO2 and the production of sugar. So this is really where the whole point of photosynthesis takes place, to make sugar. But we need energy from the light reactions to fuel the Calvin cycle to ultimately make that sugar. Now the Calvin cycle is another example of a multi-step process, and we're actually going to summarize it into 3 major phases. For each phase, you need to know what comes in and what comes out, and also where those energy molecules are being utilized. So let's start with phase 1, carbon fixation. In carbon fixation, the whole point is to take inorganic atmospheric carbon and convert it into organic carbon. So it starts with carbon dioxide and it ends with a molecule called 3-phosphoglycerate, which can also be abbreviated as 3-PGA. For each revolution of the Calvin cycle, 3 molecules of CO2 enter at one time. And if you look at what's happening just before the CO2 come in, there's a molecule called ribulose biphosphate. It's a 5-carbon compound with phosphate groups on both ends. This is the CO2 acceptor molecule. So the CO2 gets added to ribulose biphosphate, becoming a 6-carbon compound, which is short-lived, meaning that it's not stable, and it quickly breaks into 2 molecules of 3-phosphoglycerate, which is a 3-carbon compound. If you're counting carbons, there's 18 carbons here in 3-phosphoglycerate, and we will keep track of carbons as we continue to go through the cycle. Just a kind of side note that will be relevant for a later discussion when we talk about different kinds of plants based on their photosynthetic strategies. This particular version of the Calvin cycle applies to C3 plants. C3 plants are plants like rice, wheat, soybeans, and they're called C3 plants because of this molecule 3-phosphoglycerate. It's a 3-carbon compound, and it's the first organic compound that results from carbon fixation. C4 plants have a different compound that actually has 4 carbons, hence they're called C4 plants. Again, that's a topic that we'll dive into deeper at a later time, but just wanted to mention the relevancy there and why they're called C3 plants, in case you've heard that term. All right, so this is the carbon fixation phase. Now let's get to the next phase, reduction. Recall from when we talked about redox reaction, reduction is gain of electrons. So where are those electrons coming from? Well, we've got the ATP, I'm sorry, not the ATP. NADPH is our electron carrier from the light-dependent reactions, bringing those electrons to the intermediary here between 3-phosphoglycerate and ultimately the output sugar, the sugar that's produced, glyceraldehyde 3-phosphate or G3P. Now this process requires energy, and that energy is supplied by ATP. So there's going to be 6 ATP, 1 for each of the 3 PGA molecules. There were 6 of those. 6 NADPH, again, 1 for every molecule. And then we end up with 6 G3P molecules. Now this is the whole point here. This is a building block for a sugar such as glucose. So but it's interesting because not all of those sugar building blocks are available for the plant to use for a source of food. In fact, only one of those 6 G3Ps leaves the Calvin cycle. The other G3Ps, which there'd be 5, will remain in the Calvin cycle for the third stage. The third stage is the regeneration of the CO2 acceptor ribulose biphosphate, or RuBP. This needs to be regenerated because in order for the Calvin cycle to continue, there has to be a molecule that can receive the carbon dioxide to ultimately fix it into organic carbon. So 5G3P get phosphorylated. There's several intermediary steps here, resulting in 3 molecules of ribulose biphosphate, 1 for each CO2 that enters the next round of the cycle. Let's go back and just count the carbons again and make sure we haven't lost any. So at the end of reduction, there were 6 times 3 or 18 carbons, but we lost 3. They exited the cycle as a G3P. So that means we have remaining 15 carbons, and that works out here. 5 of the G3P that go back into the cycle represent the 15 carbons. And even after these several steps that occur, including phosphorylation from ATP, there's going to be only 3 molecules of ribulose biphosphate, but it's got 5 carbons. So 3 times 5 is also 15 carbons. All right, and it continues to be 15 carbons until 3 more carbons come in from the CO2. And now we're back to 3 times 6, 18 carbons. Keeping track of the carbons is helpful because it might help you remember where carbons enter and exit the cycle. And something else interesting to notice is that there's 3 carbons coming in the form of CO2, and there's going to be 3 carbons leaving as G3P. So it's balanced out. And the rest of them, the other 18, remain in the cycle. Okay, so in summary for the Calvin cycle, yeah, there's a lot going on. The biggest take-homes here are that you need to know how many ATP and NADPH are consumed for each G3P that's made. There's going to be 9 ATP, 6 NADPH, and the whole point, though, is to make that sugar G3P. So what's going to happen to this G3P? Well, there's a few different fates here for it. One possible fate is that it's going to be converted to glucose or fructose, which both have the same molecular formula, making them isomers. That formula is C6H12O6. The purpose of doing that is that it's now an immediate source of energy for cellular respiration. It can also be used as a building block material for other molecules. Another possible fate of the G3P is it can first be converted to glucose or fructose, and then to starch, which is the storage molecule in a plant for excess food. This is particularly important for plants to have at night when photosynthesis is not taking place, but they still need to have a source of food to continue doing work and surviving.